Hello students, welcome to EPG Pathshala. I am Dr. Vishvanath Tiwari from Central University of Rajasthan. Today I am going to talk about basic of enzyme inhibition. This module has been taken from a paper Molecular Enzymology and Protein Engineering. In this module, we will discuss about basics of enzyme inhibition. So this module has been divided into the following components. First one is the introduction about what is enzyme inhibition, and then different types of enzyme inhibition such as irreversible inhibition, suicide inhibition, reversible inhibition, substrate inhibition and product inhibition. We will also discuss about the different types of these inhibitions. So first of all, let's ask a question, what is inhibitors? So the molecule that binds to the enzyme and reduce the rate or activity of enzyme catalysis is known as enzyme inhibitor. How this enzyme is going to inhibited by the inhibitors? So there may be following mechanism an inhibitor can adopt to create the enzyme inhibition. The first one is binding of inhibitor might alter the binding of the substrate. Therefore, they can inhibit the enzyme. The second thing is inhibitor may modify the significant amino acid residues in the active site of the enzyme. As you know that active site amino acids are playing significant role in the catalysis. If they are covalently modified by the inhibitor, then catalysis get inhibited. And the binding of inhibitor is either reversible or irreversible. Usually two type of interaction takes place between all the biomolecule and its ligand. The first one is covalent interaction, where a permanent bond is formed between enzyme, substrate, enzyme, inhibitor. The second one is a non-covalent interaction. Non-covalent interaction are weak interaction where no covalent bonds are formed. A specificity and selectivity of a drug is very important to create a effective enzyme inhibition, and it is important for selecting a particular drug or drug of choice. The first of all, enzyme affinity with the inhibitor. So the good interaction will enhance the efficacy of the drug. So different kinetic parameters like Km when you are talking about enzyme substrate, Ki when you are talking about the inhibitor and enzyme are important for binding affinity of a particular drug to its enzyme. Many non-covalent forces such as hydrophobic forces, van der Waals forces, hydrogen bonding and others are responsible for the binding affinity. A specificity of a drug will be decided by the number of directional forces. The important point is that what are directional interaction, what are non-directional interaction. Directional interaction includes ionic bonded, hydrogen bonds, while non-directional forces involve hydrophobic interaction and somewhat van der Waals forces. So a specificity of particular drug is very important. If a drug is specific and that is going to work on a specific enzyme, because when you are talking about taking a drug from outside, the body is having n number of enzymes. So a particular drug is going to bind to a particular protein, particular enzyme, that will be decided by the selectivity or a specificity of a particular drug. If the drug is less specific, then it may cause some side effects as well as toxicity to the human who is taking that drug. Enzyme inhibitor are not only synthesized chemically, but it can be isolated from the natural sources which inhibits metabolic pathways, and feedback inhibitor is a well known inhibition where the inhibitor is isolated from the biological system, because in the feedback inhibition it is a product that inhibits its own production. So that is a naturally isolated inhibitor. So enzyme inhibitor play a very significant role in controlling the excessive activity of enzymes like proteases, which are may cause some lethal effect to the human being. So different types of inhibitor are there. The first inhibitor is one that have the mechanism by irreversible inhibition, where the reversibility of the inhibition is not there. That means once the inhibition is done, you cannot reverse it. Then suicide inhibitor, it is a specific case of covalent modification inhibitor, but that is also known as mechanism based inhibition. Then third one is reversible inhibition. This mechanism is most diverse and well studied mechanism. Reversible inhibition, the inhibition can be reversed by some means, and that is subdivided into the competitive inhibition, non-competitive inhibition, uncompetitive inhibition, and mixed inhibition. Other mechanisms of inhibition are feedback inhibition. The feedback inhibition is very significant when we are talking about the metabolic regulation of different pathway in the biological system. And then last one is the substrate inhibition. Sometimes substrate is also inhibiting the enzyme activity. So we'll discuss all these into detail in this module. Irreversible inhibition, as the name suggests, you cannot reverse this inhibition. It is based on the formation of covalent bond between enzyme and the inhibitor. This covalent bond, which is formed between enzyme and inhibitor, is not reversible. Therefore, the EI complex or enzyme inhibitor complex, you can say, is a dead end complex where we don't have any product formation. The mechanism of these inhibitors is based on how they are going to modify the amino acid residues that is present in the active site of the enzyme. Therefore, it is possible the presence of a particular functional group in the active site is important for producing mechanism of action of a particular irreversible inhibitor, which undergoes a specific covalent modification. It is known that active site residues have a side chain which is either electrophile or neutrophile. So if side chain of active site is electrophile, then it will interact with the nucleophile groups. So nucleophile group must be present in the irreversible inhibitor when it will inhibiting a electrophile group containing active sites, and vice versa. For example, if your active site have nucleophile group, then your irreversible inhibition must have electrophile groups. For example, lysine, you know that it will have the positive charge epsilon amino group. It will interact with the nucleophile by forming covalent eject, and well-known CIF base is one of them. The second one is hydroxyl group, the serine and threonine and tyrosine, which is going to have negative charge oxygen. Similarly, sulfahydryl group of the cysteine, they all are nucleophile, so they interact with the electrophile. So if you want to design a drug against the lysine of an active site, you must to have nucleophile in the inhibitor. If you design a drug with a serine or other nucleophile containing active site, the molecule must have electrophile groups. So we have few examples of irreversible inhibitor, and well known, few of them is very well known. The first one is DIPF. As I already told you that the serine is very important in the active site of some enzyme, and that is serine proteases and acetylcholinesterase. So if you are having an inhibitor that binds with the serine covalently, it will irreversibly inhibit these enzymes. The second one is iodoacetate, which covalently modify the sulfahydryl group of the cysteine in the active site. Then it will act as an inhibitor, irreversible inhibitor for the enzyme that uses sulfahydryl group. One such example of that enzyme is papain. The penicillin, if you remember penicillin, it is a first antibiotic that is discovered by Alexander Fleming. It's also based on the irreversible inhibition mechanism, and it will bind to the serine of penicillin binding protein. Another inhibitor is a ritonavir, which is a reversible inhibitor of HIV protease. Suicide inhibition. Suicide inhibitors are itself is not an inhibitor. They are non-toxic compound, but they are converted into the product by a particular enzyme of your biological system. So, suicide inhibitor itself is considered as a product drug or pro-inhibitor rather than inhibitor. But because of action of the enzyme, suicide inhibitor is activated, and once the suicide inhibitor activated, it irreversibly inhibiting the enzyme. Therefore, you can also say that suicide inhibitor are mechanism based inhibitors. For example, DFMO, that is difluoromethylornithine, is a suicide inhibitor of ornithine decarboxylases, which is involved in the polyamine biosynthesis. DFMO uses by ornithine decarboxylase in the place of its own substrate, that is ornithine, and undergoes decarboxylation. So after decarboxylation, it is activated and then it is irreversibly inhibiting the ornithine decarboxylase. DFMO is used as a treatment for sleeping sickness. Reversible inhibition. As the name suggests, you can reverse this inhibition. So when you are talking about reversibility, that means we don't have any permanent interaction between reversible inhibitor and your enzyme. Therefore, most of the interaction that is taking place between reversible inhibitor and enzyme is governed by weak non-covalent interaction, which includes hydrogen bonding, hydrophobic interaction, van der Waals forces, and Salt bridges. So the cumulative effect of these interactions result into the strong interaction. But because of these interactions are weak reversible interaction, so you can reverse the process of inhibition. On the basis of varying concentration of enzymes substrate on the inhibitors, the reversible inhibition can be grouped into four groups. The first one is competitive inhibition, where the structure homology between substrate and inhibitor. The second is non-competitive inhibition, and third is uncompetitive inhibition, and fourth one is mixed inhibition. So we will discuss about all these four types of inhibition one by one. Reversible inhibition can be quantitatively described by inhibitor concentration required for binding to the enzyme and producing enzyme inhibitor complex, and that constant is known as Ki, or catalytic constant for inhibitor. Similar to the Km, if the Ki is low, then a particular inhibitor have better interaction between enzyme and inhibitor, and Ki is accurately determined by isothermal calorimeter, where the titration of enzymes with the inhibitor can be done, or by other methods like Michaelis-Menten equations. In this way, we have learned that how reversible inhibition is used for analysis of a particular type of inhibition. This is a first type of reversible inhibition, where the competitive inhibitors are structurally similar to the substrate, therefore they will compete for binding to the active site of the enzyme. Therefore, the binding site of active substrate as well as competitive inhibitor are common, So both will bind to the active site, and based on the binding affinity of a competitive inhibitor or the substrate, the candidate which will bind to the active site will be decided. Therefore, at a given time, free enzyme can either form ES complex with the substrate or produce EI complex with the inhibitor, but we will never have formation of ternary complex like ESI complex. If competitive inhibitor have better interaction or low Km, then more competitive inhibition take place, while if substrate have better interaction or low Km, then less inhibition take place. Competitive inhibition result into the increase in the Km of the substrate. That means they are altering the binding of the substrate, but they will not have any effect on the Vmax. So we will discuss what is the basis of increase in the Km, why Vmax doesn't change at all in our next module. So the competitive inhibition can be reversed by increasing substrate concentration. When we are talking about drug designing based on the competitive inhibition, then it is important to know that how your enzyme is involved in converting substrate into the product. So substrate will bind to the active site of the enzyme and convert it into transition state, and this transition state is converted into the product. And studies shows that transition state have a structure that is different from substrate and product, but they have the highest binding affinity with the enzyme. Therefore, if you are designing a competitive inhibitor which mimicking transition state, it is going to be a better inhibitor as compared to designing an inhibitor which is competing the substrate. Therefore, the transition state analogs are designed which is having lower Ki, even more affinity with the inhibitor designed against the substrate. One such examples are is oseltamivir. This is mimicking the oxonium ion of transition state of the reaction catalyzed by neuraminidase. So therefore, this model of designing inhibitor against the transition state is going to be a very promising approach to develop new drugs. So we'll discuss about examples of competitive inhibition. The first one is a methanol. It is well known when it is taken along with the ethanol. So methanol is a competitive inhibitor of alcohol dehydrogenase, which is responsible for converting ethanol into acetaldehyde. There is a structure homology between methanol and ethanol, therefore there is a competitive inhibition. The second one is a malonate. This malonate have structural homology with the succinate, therefore they act as a competitive inhibitor for succinate dehydrogenase that converts succinate into the fumarate. The another examples are methotrexate and aminopterin. They are competitive inhibitor of an enzyme that is known as dihydrofolate reductase. This is important enzyme when we are talking about the nucleotide biosynthesis. Therefore, they are sometimes used as an anti-cancer drug. The another example of competitive inhibition is a statin drug. Statin group of drug have structural homology with the HMG coenzyme A, which is a substrate of HMG coenzyme reductase, and that is very important when you are talking about the cholesterol biosynthesis. Therefore, statin drug has been taken as a anti-cholesterol drug. And last one is a sulfur drug that is also act by competitive mechanism. After discussion of competitive inhibition, the next type of reversible inhibition is non-competitive inhibition. As the name suggests, non-competitive inhibitor does not have any structural resemblance with the substrate. Therefore, they doesn't have common binding sites. As you can see that substrate bind to the active site while non-competitive inhibitor bind to the non-competitive inhibitor binding site. Please don't confuse non-competitive inhibitor binding site with allosteric site because they have different properties. So the binding of non-competitive inhibitor and the enzyme are different. Therefore, they will not alter the binding of each other, so the Km remains same. But because the formation of ESI complex takes place when inhibitor binds, hence the Vmax will be altered. It is important to know that non-competitive inhibitor can bind to the free enzyme or to the ES complex. It is very important to note that sometimes some unique mechanism takes place where ESI complex, which is most of the time think that it will not form any product, is going to be catalytically active. And once it is catalytically active, it is either having lower catalytic activity, then it is known as partial non-competitive inhibition. If it is having higher catalytic activity, then it is known as partial non-competitive activation to the ES complex. Non-competitive inhibitor are one that will does not affect the ES formation, but it will affect the product formation. One such examples are binding of ciprofloxacin or nalidixic acid to the DNA gyrase. After discussion with the competitive and non-competitive inhibition, the third category is uncompetitive inhibition. The uncompetitive inhibitor are similar to the non-competitive inhibitor, but the difference is uncompetitive inhibitor will never bind to the free enzyme. It will only bind to the ES complex. Therefore, you can say that binding of uncompetitive inhibitor and substrate are dependent to each other, but the dependency is one side. For example, uncompetitive inhibitor will only bind when substrate is there on the active side, but substrate will bind when we don't have uncompetitive inhibitor to the active side. In this way, because the binding of uncompetitive inhibitor is influenced by the substrate, so the Km will alter. At the same time, ES complex which is formed will not form product, therefore Vmax will also alter. What is the reason of this uncompetitive inhibitor to alter the Km? If you remember the Leach-Atelier principle, here we have enzyme combined with a substrate producing ES complex, and ES complex is going to be bind with the inhibitor, uncompetitive inhibitor, and converted into ESI complex. So Leach-Atelier principle indicating that any reaction will shift toward that side where either reactant or product is consumed. So in this way, if your inhibitor is continuously binding to the ES complex, they are shifting equilibrium toward the formation of more ES complex, and it is only possible when affinity between your inhibitor and your substrate are different, but in the presence of inhibitor, your affinity of substrate will increase, hence Km will decreases. After discussion of three type of reversible inhibition, the fourth one is mixed inhibition. If you remember non-competitive inhibition, where both binding of inhibitor and substrate are independent, in uncompetitive, inhibitor is dependent on the binding of substrate, but substrate is independent to the binding of inhibitor. In the mixed inhibition, this type of non-competitive inhibition where the binding of substrate is dependent on the binding of inhibitor, as well as binding of inhibitor is also dependent on the binding of substrate. So based on the type of mixed inhibition, it is either competitive non-competitive inhibition or uncompetitive non-competitive inhibition. Both are having different kinetic variations. For example, in competitive and non-competitive inhibition, Km increases and Vmax decreases, while in uncompetitive non-competitive inhibition, Km decreases and Vmax also decreases. The next type of inhibition is feedback inhibition or product inhibition. The feedback inhibition is the name given to those inhibition where product itself inhibits its own formation. This is important when we are talking about the biological reactions where a substrate is converted into the product and product is utilized in the next cycle. So if you have excessive product remain in the biological system, then you doesn't require that particular product to be formed. Therefore, that product itself inhibits its own production, and it is either branch feedback inhibition, either sequential feedback inhibition, either concerted feedback inhibition, either cooperative feedback inhibition. So this all will be discussed in the next module, but it is very important in terms of regulating biological activity. Next type of inhibition is substrate inhibition. Usually substrate is not acting as an inhibitor for the enzyme, but sometimes if we have excessive substrate, an enzyme is following the two substrate binding model, then it may act as a inhibitor, but it always act as inhibitor when we will have higher substrate concentration. So at lower substrate concentration, one substrate bind to the active site and then it has good activity, but at higher substrate concentration, the second site, which is designed for binding to the subsequent molecules, is occupied by the same substrate, and this second site act as a inhibitory sites, which decreases the enzyme activity, and this mechanism is known as substrate inhibition. Although this is a rarely occurring inhibition, but it is very interesting to know that substrate can act as an inhibitor. So in this discussion, we have discussed about competitive inhibition, where the drug designing is going to be take place around the transition state, then non-competitive inhibition, uncompetitive inhibition, and feedback inhibition and substrate inhibition. So if you compare feedback inhibition and the substrate inhibition, there are two different aspects of it. So product, when it acts as an inhibitor, it is known as feedback inhibition, but when substrate acts as an inhibitor, is known as substrate inhibition. So feedback inhibition have more biological significances because they are basically preventing excessive substrate to be used. But here, if your concentration of substrate is higher, then it will itself act as an inhibitor. So it is very interesting to know that in the case where two state binding take place, then feedback inhibition itself promoting the substrate inhibition. So therefore, for normal reaction, feedback inhibition is better. So it is preventing excessive substrate to be waste. But at the same time, in the case of two substrate bonding model, the feedback inhibition might be a problem for substrate inhibition, but this study is not yet proved, so it is going on. So student, let's now summarize what we have learned from this module. The first thing we have learned what are enzyme inhibitor, and then we have learned what are the different type of inhibitors. The first one, we have learned about details of the competitive inhibition, non-competitive inhibition, uncompetitive inhibition, and mixed inhibition. Reversible, they all belong to reversible inhibition, and reversible inhibition is very significant when you are talking about the drug designing. We have number of drugs that are designed against a particular enzyme using the competitive inhibition model. An interaction between enzyme and inhibitor play a very significant role in enzyme inhibition. So therefore, it is important to study what are the dissociation constant of an enzyme with a particular inhibitor, how a particular inhibitor is going to interact with the particular enzyme to exert its action. So we have also discussed about irreversible inhibition, where the reversibility is not there. So when we are talking about the irreversible inhibition, they are very lethal. So you cannot have any option if the irreversible inhibition take place. Then we have discussed about suicide inhibition, where the inhibitor is itself a prodrug or pro-inhibitor, which is not an inhibitor, but because of mechanism based conversion, that prodrug is converted into active inhibitor, so they will inhibit a particular enzyme. At the same time, we have discussed about feedback inhibition. So in feedback inhibition, the product itself inhibits conversion of substrate into product. So that means if you have excessive product form, that will basically block its own conversion and preventing the substrate to be utilized in other mechanism. So in this way, we have learned how the enzyme inhibition is playing a significant role in terms of drug designing, in terms of studying the metabolic pathways. Thank you.